Hello everyone, welcome back to Biology Board. This is Brijesh Chotaliya and today we will study about DNA repair. We will here discuss about how it is damaged, then how it got repaired. We will study about several DNA repair mechanisms. So let's start with our today's lecture. DNA repair. DNA repair, that is, damaged DNA must be repaired. If the damage is passed on the subsequent generation, then we use the evolutionary term mutation. It must take place in the germ cell, the gametes, eggs, and the sperm. What are gametes? Gametes are female eggs and male sperms. Why here it is taken that must take place in germ cell? Because if the damaged DNA is passed on the subsequent generation, then the mutated species will grow. Okay. If damage is to somatic cells, then just that one individual is affected. Okay. If the damage is in the somatic cell, then only the person in which the DNA is damaged, that one individual person will get affected. But if the damage is happened in the gametes, that is in germ cells, then it is more possibility that the next generation will be the mutated generation. One of the main objectives of biological system is to maintain base sequences of DNA from one generation to another. Changes in the DNA sequence arise during replication of DNA damage by chemical mutations and radiations. During replication, if incorrect nucleotides have been added, they are corrected through editing system by DNA polymerase 1 and DNA polymerase 3. So let's say that this is DNA. These are the nucleotides arranged in line. Okay. So in replication, if incorrect nucleotides are arranged, then they are corrected by DNA polymerase 1 and DNA polymerase 3. The other systems also exist for correcting the errors missed by editing function. It is called mismatch repair system. We will study about in detail about this one. Okay. Mismatch repair system edits the errors left by DNA polymerase 1 and DNA 3 and removes the wrong nucleotides. So, if incorrect nucleotides is also added by this DNA polymerase 1 and DNA polymerase 3, then this system, mismatch repair system, it edits the leftover errors of DNA polymerase 1 and DNA polymerase 3. Then again, proofreading is done by polymerase 1 and 3. Okay. DNA is always damaged and mutated by several chemicals and radiations. Of course, chemicals and radiations will do several damages to DNA, and the mutations are at higher risk. Only a few errors accumulate in DNA sequence. The stable errors cause mutations, and the rest is eliminated. This is DNA. These are the damages in the DNA. Okay. Most probably the damaged DNA is solved, but there are some errors which cannot be solved, and these errors causes mutations because these errors become stables and can't be this error cannot be solved. Okay, these stable errors caused mutations, and the problem is not solved, and due to this, the mutations occur. If errors in DNA sequence are corrected before cell division, no mutations occur. Yes, if the errors in the DNA sequence are corrected before cell divisions, during cell divisions, okay, it must be corrected. If not corrected, then mutations occur. However, there are some DNA damages which cannot be mutated because the damages are not replicated. Okay, remember this point. Therefore, such damages causes cell death. What is cell death? Apoptosis. So, how the DNA is damaged? There are four most important points. Consequences of DNA replication error, that is damage caused during DNA replication, chemical agents acting on the DNA, and UV light imparting energy into DNA molecule, that is ultraviolet radiations. Due to ultraviolet radiations, DNA got damaged. and the spontaneous changes to the DNA due to some changes in DNA. These are the four main objectives of DNA damage. There are several types of damages that occur in DNA. What is first? Modifications of one or more bases by highly reactive chemicals such as alkylating agents. Then losses of purine base due to local pH change. We call it as a depurination. We have already discussed this about in DNA damage lecture. Okay, if uh, if the person who is watching this video does not know about this, can go to my channel biologyboard and see the previous lecture DNA damage. You will get these all points there. Then the third one is single strand or double strand break due to the bending or shear force. Then dimer formation, that is TT. We have talked about there pyrimidine, two adjacent pyrimidine molecules due to ultraviolet and extra radiations. What happens due to dimerization? No hydrogen bond with opposite purine shall occur. Okay, this results in distortion of helix. Most of the spontaneous errors are temporary because they are soon created by a process called DNA repair. So these are the types of DNA. We have covered the most important DNA damages and its types in the previous lecture. Okay, so DNA repair mechanisms: photoreactivation, base excision repair, nucleotide excision repair, mismatch repair, double strand break repair, homology directed repair, and source response. These are the DNA repair mechanisms in DNA repair. Okay, photoreactivation. The UV damages caused in cells are repaired after exposures of cells in visible light. This is called photoreactivation. How does it take place? The DNA is damaged by a ultraviolet radiation, and the damaged DNA is recreated by the exposure of, that is repaired by visible light, and this process is called as photoreactivation. In this mechanism, an enzyme DNA photolyase cleaves TT dimers and reverts to monomeric state. Okay, this is a TT dimer here. This is a photolyase enzyme. It cleaves the TT dimer. Here, the photolyase enzyme, it cleaves TT dimer. The bond between TT, that is adjacent pyrimidine bases. The bond is created between adjacent pyrimidine bases instead of opposite purine base. Okay. This enzyme is activated only when exposed to visible light. That is this DNA photolyase. The mutant cells lack photolyase. This enzyme absorbs energy, binds to cyclobutene ring to defective sites of DNA, and promotes cleavage of covalent bonds formed between TT. This enzyme is found in several bacteria and placental mammals. Finally, thymine residues are made free and damaged is repaired. In short, this is a TT dimer, okay? Photolyase enzyme, in the presence of visible light, it breaks down the bond between this TT and the cleavage of cyclobutene ring. This ring cleavage, the cleavage part is then repaired. Here it is repaired, and the photolyase enzyme is released, and TT dimer also released. Due to this photoreactivation, for this figure shows reactivation for repair of thymine dimer. Base excision repair. The regions containing non-helix distortion are repaired by base excision repair. It involves at least 6 enzymes called DNA glycolases. Each enzyme recognizes at least bases and removes from DNA strand. The enzymes remove deaminated cytosine, deaminated adenine, alkylated or oxidized base. Base excision repair pathways start with a DNA glycosylation. For example, the enzyme uracil DNA glycosylase removes the uracil that has wrongly joined with G which is really deaminated cytosine. We are here talking about the damage caused by deamination. In previous lecture, that is in DNA damage, we talk about deamination. It is a type of DNA damage. So base excision repair is the type of repair which helps in this type of DNA damages. Then AP endonuclease and phosphodiesterase removes the sugar phosphate. AP sites arise as a result of loss of a purine or a pyrimidine. A gap of single nucleotide develops on DNA, which acts as a template primer for DNA polymerase to synthesize DNA and fill the gap by DNA ligase. So we will understand here this is base excision repair and this one was the nucleotide repair. We will study about in the next few minutes. So this is DNA. In the presence of C, here it goes to deaminated and instead of C, here the presence of U, that is uracil. Now G cannot get binded with uracil. Okay. Then uracil DNA glycosylase comes and it removes U. Okay. AP endonuclease and phosphodiesterase remove that part of U, that is uracil, a gap is formed, then C is added. Therefore, the damage is repaired, that is C and G. This is base excision repair. Uracil is removed by uracil DNA glycosylase, AP endonuclease and phosphodiesterase diesterase removes that part and the gap is formed and the gap is filled. Okay, and this is base excision repair. What happens in nucleotide excision repair? Any type of damage having a large change in the DNA helix causing helical changes in DNA structure is repaired by this pathway. Such DNA damage may arise due to pyrimidine dimers, that is TT, TC, CC, caused by sunlight or covalently joins large hydrocarbon. In E. coli, a repair endonuclease recognizes the distortion produced by TT dimer and makes two cuts in the sugar phosphate backbone on each side of the damage. The enzyme DNA helicases removes oligonucleotide from the double helix containing damage. DNA polymerase III and DNA ligase repair the gap produced in the DNA helix. So what happens? This is one nucleotide. Here a dimer is formed. Instead of joining with this, it creates bond between this T and C. Okay. Then a part of DNA is cut. That is, we say it as a oligonucleotide is cut and removed. Then DNA polymerase and ligase joins the gaps here. Okay. And the DNA is repaired. This full DNA is repaired. So this is damage repair. This is base excision repair and this one is nucleotide repair. Only the difference is here oligonucleotide, that is we say about 8 to 10 base pairs are removed instead of this dimer. 8 to 10 base pairs are removed at a time, but here only a single, in base excision pair, a single base is cleaved and a gap is formed. Okay. So in all organisms, NER involves the following steps. What is NER? Nucleotide excision repair. First, damage is recognized. Then binding of a multi-protein complex at the damaged site. Double incision of the damaged several several nucleotides away from the damaged site on both the 5' and 3' sites. Then removal of damage containing oligonucleotide from between the two nicks and filling in of the resulting gap by DNA polymerase and then ligation. Okay, so everyone must be clear with these terms. Now we will study about methylation directed very short patch repair. We can call it as mismatch repair also. Okay, so this repair is accomplished by involving methylation of bases, especially cytosine and adenine. In E. coli, methylation of adenine and in sequence of GATC is done by the enzyme methylase, a product of DAM gene on both strands of DNA. What is GATC? It's a sequence which is recognized during the methylation of adenine process. Okay. So after replication, only A, that is adenine of GATC of one strand remains methylated while the other remains unmethylated until methylase A accomplishes methylation. So here we can see this here. This A, it gets methylated. Okay. Then, in E. coli, repairing activity requires four proteins. What are the four proteins? Mut L, Mut S, Mut U, and Mut H. And it is redirected by the genes Mut L by Mut L, S by Mut S, Mut U by Mut U, Mut H by Mut H, respectively. Okay. The Mut genes are the loci which increase the frequency of spontaneous mutation. The unmethylated GATC allows the mut L to recognize the mismatch during transition period. We can see in the figure, mut L, it recognizes the site. See here, the bond is formed between G and T. This should be repaired. G always binds with C and T always binds with A. So here a repair must be done. So mut L, it recognizes the damage. Then this helps mut S to bind to the mismatch. When the mut L finds the damage, mut S get bind to the damaged site. That is mismatch pair. Okay. Mut U supports in unwinding the single strand and single strand DNA binding proteins and maintains the structural topography of single strand. Mut H cleaves the newly synthesized DNA strand and the protein Mut U separates the mismatched strand. So Mut U, it helps in unwinding and separating the mismatch pair, and Mut H cleaves the newly synthesized DNA. Mut H, what does it do? Mut H, it cleaves this DNA. Okay. However, there is a gradient of methylation along the newly synthesized strand. Least methylation occurs at the replication core. The parental strand is uniformly methylated. The methylated bases direct the excision mechanisms to the newly synthesized strand containing the incorrect nucleotides. During this transition period, the repair system works and distinguish the old and new strands and repair only the new strands. So, we will shortly discuss what is the message they are conveying to us. Okay, mismatch repair. This is a type of mismatch repair only. This is methylated adenine. Okay, the adenine is methylated, but here is mismatch. G get bind with T instead of C. So mut L, it recognizes the mismatch. Mut S, it binds to the mismatch here. Okay. What does mut H do? It cleaves the part of mismatch, and mut U, it stabilizes. What they are saying, it separates and unwinds. It separates and unwinds the DNA. The function of mut U. Then replacement of correct base. Then the base is corrected here. Let's see here. It was G and T. Then the corrected one G and C. So what is the figure one? Excision of a newly synthesized strand and repair system. What does figure B shows? It shows arrows shows the region where methylation is not complete. This arrows shows methylation is not complete, and the dark region line shows the region where methylation is complete. This region, this shows where methylation is complete. That is in parental strands. Okay. This is about mismatch repair. Then the next one is recombination repair. That is daughter strand gap repair. When excision repair mechanisms fail, this mechanism is required to repair errors. This mechanism operates in the viral chromosome in host cell whose DNA is damaged. This mechanism operates only after replication. Therefore, it is also known as post-replication repair. Post-replication repair. Remember this term. Probably Rec A protein in E. coli catalyzes DNA strand for sister strand exchange. Thus, a single-stranded DNA segment without any defect is excised from a strand on the homologous DNA segment at the replication fork. Okay, it is inserted into the gap created by excision of thymine dimers. Then the combined action of DNA polymerase I and DNA ligase joins the inserted piece. The gap formed in donor DNA molecule is also filled by DNA polymerase I and ligase enzyme. Okay, so this is a recombination repair mechanism. What does it do? A dimer present over here, a replication fork is created. So dimer was present here, okay? A DNA segment got excised, a gap is formed, okay, and then DNA polymerase 1 and DNA ligase, DNA polymerase brings about the replication of this gap region, and DNA ligase joins with this remaining DNA. This is recombination. Only the dimer is removed. The main function of this is recombination repair. Okay, gap repair. We can say this as daughter strand gap repair. Then the next one is source repair. What is source? Save our soul. Save our soul. Repair is a bypass repair system. It is also called emergency repair. This repair is not as functional as the previous we have studied, but let's study about this source repair also. The damage in DNA itself induces the source regulator system, which is a complex cellular mechanism. SOS works where photodimers are formed that lead to the cell death. SOS is the last attempt to minimize mutations for survival. It induces a number of DNA repair processes. SOS system works in the absence of DNA template. In the absence of DNA template, it only works. Okay. Therefore, many errors arise leading to mutation. Generally, genes of SOS system remains in repressed condition caused by a protein LEX-A. Repression of SOS is inactivated by REC-A protease. It is formed after the conversion of REC-A protein by DNA damage. Okay. So, DNA damage results in conversion of REC-A protein into REC-A protease. Let's understand here about in the figure. Okay. This is whole mechanism of SOS repair system. What they are saying first, DNA damage results in conversion of REC-A protein into REC-A protease. REC-A protein get converted into REC-A protease. Step 1A. Sorry. It is 1. Okay, step 1. Then REC-A protease breaks LEX-A protein. This REC-A protease breaks LEX-A protein. Step B. Normally, LEX-A protein inhibits the activity or REC-A gene. Inhibits activity. REC-A. The DNA repair genes UVRA and UMUD. Step D. Okay. UVR protein, UMU protein, and other source genes. Okay. Clear about? Finally, DNA repair genes are activated. After this process, DNA repair genes are activated. SOS system does not repair the large amount of DNA. As I have told earlier, SOS system is not very much functional. When DNA repair is over, REC-A protein loses its proteolytic activity. Then LEX-A protein accumulates and binds to SOS operator and turns off SOS operator. Okay. However, repression is not complete. Besides, some REC-A protein is also reproduced that inactivates LEX-A protein. Okay. So this is the old schematic diagram, and it shows mechanisms of SOS repair system. So this is all about our DNA repair system, DNA repair mechanisms. Okay. If you have any doubt, you can comment us. The comment section is open for everyone. You can kindly tell us about our interactions with you. Okay. If you have any doubt, you can ask in the comment section below. Okay. So stay tuned for more videos. Hope you like the video. Okay. Thank you so much for watching this. Thank you.